My name's Rodney. You may have heard about me through Jeff. So Jeff and I had met about 11 months ago. It was very interesting. And I'll tell you about that story in a bit, but a little bit about what my talk is about. Originally, it was supposed to be about the mouse models and only about the mouse model update, right? Then as I sat through the talks this morning and I was watching your faces, yes, I was watching you from over there, and I saw that perhaps we were not all on the same page in terms of what some of these terms meant when we say phenotype or we say mouse or rat or zebrafish or iPSC, blah, 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 right? So I'm from a military family, so I talk to my dad a lot, right? And I tell him about what I do, and he's always asking me, what are you talking about? So I'm very used to kind of making it more for a lay audience in terms of trying to articulate what we as scientists or physician scientists are trying to attempt in the laboratory and bring that from the lab to the clinic, but then also engage the community to understand what exactly is going on with your condition or your child's condition, and then try to bring it back to the lab and make it an iterative process, right? So I'm at Baylor College of Medicine in Houston, Texas. I don't know if you could see that, but it's down there, the map of Texas, not Baylor College of not Baylor as in Baylor-Waco, which is in Dallas. And I am affiliated with the Texas Children's Hospital, which is the largest medical center in the US, well, children's hospital as well, and has more than 106,000 employees, 10 million patients per year. So it's a pretty cool place to work. You could see there to the right, that's what it looks like at night. That's the NRI, and that's my team below. So it really takes a village and a community to make this work, right? So you've heard the phrase, teamwork makes the dream work, and I think that really has been the bottom line with a lot of the discussion today. As much as we are so proud of Jeff and we applaud him for everything that he's done, he's only one person, right? And I think for Stacy too, only one person. It really requires the entire set of families and their relatives and researchers to work together to get, I would say, actionable therapy in place. I hope that makes sense. And interrupt me anytime during this conversation if you'd like. So, and that teamwork is important because there are 7,000, as we know now, but I put that plus because there probably are even more rare genetic disorders. And in orange, collaboration, communication, and engagement, the voice of the individual, that patient voice is key. It's becoming more important to the FDA these days. So by coming together and bringing your stories and compiling that, going to the FDA and being ready when a therapy is available will be crucial to making sure that Champ1 is at the top of that list of 7,000, right? So that bridge here, where I work, is that translational arc. So I say an arc, or bridge, et cetera, whatever you want to call it. I'm a basic scientist, so I was trained as a geneticist, but I work with mice, and I work with their brains, so I'm also a neuroscientist. But in the recent years, what I've learned is that a lot of the basic biology that we do, that really isn't, I would say, enhancing the lives of people who are suffering from these conditions. What is really needed is understanding how we take those tools, that biology, and transform them into those therapies. And that's where the preclinical work comes into place. And by doing so, we work with the community and advocacy groups to push it forward, right? So that's that bridge there, and it's bi-directional. But finding common ground is also key. So a lot of that stuff that you may not be able to see down below, that's all related to research stuff, right? Basic science stuff. So on the left, those colors, those are brains that we study of mice. But on the right, what that shows is a timeline. And that timeline is a natural history study of animals, right? Of a specific genetic model, another one that I study, is exactly what we're going to have to do for Champ1 because, as you know, we don't really have the full picture of how the Champ1 progression occurs over time. And so we have these animal models to understand and possibly predict what may happen when children now may be affected with vision problems can see when they're adults, right? So this natural history study actually came from, or that approach came from natural history studies they do in people with Alzheimer's disease, with Parkinson's. So we've adopted that kind of method to better understand some of our model systems. But who, what, when, where, why, and how, those are the very simple, I would say, questions that we've learned from sixth grade education, or US education, or less than sixth grade, that we're trying to address with all of the studies that we're doing and what we're trying to address today. And keeping that in mind, moving forward, I think with the model systems, that's one aspect to ensure that we get from bench to bedside, right? So this shows basically the 4 different phases for getting, I would say, a drug approved in a very simple way, right? By the FDA, the US FDA. And that space here, that preclinical animal models, the basics, that's where we are right now with Champ1. Not a lot has been done, but we do need to move that forward, right, to clinical trials and then IND and then post-marketing. So what I think those numbers that you saw below in terms of the number of patients you would need per phase, there are new mechanisms in place for rare genetic disorders that will not require, I would say, those numbers of patients. But we could talk about that offline. So one aspect of work that Wendy had talked about, as well as Zhang, was the fact that we need these model systems, whether they be animal models or cell models, to find these biomarkers and outcome measures. So these are surrogate endpoints that we try to identify in our tools to then test a compound or test some kind of natural supplement or test gene therapy. And those surrogate endpoints may not necessarily mean that we could get the mouse to start talking again, right? It could be something as simple as the mouse moves differently when you treat it with some kind of drug compared to with no drug, right? And that it's robust enough and reproducible enough, but more importantly, there's some kind of translational relevance. So what does that mean by translational relevance and disease relevance, right? Disease relevance is that if I make a mutation, let's say if I make JJ's mutation or Lucas's mutation in a mouse, and it's the exact same mutation, right? There is some disease relevance, right? Some construct validity, that's what we call it. But then let's say we take those mice and we put it on this mat, right? Where the mice could walk along and they have abnormal gait. That's the same kind of mat that we use in the clinic, or not myself, but my collaborators would use in the clinic to test a child that has altered gait or abnormal gait. So that's the translational relevance. Can we do a test that's almost exactly the same way that we would do in the clinic? Now, at the end of the day, a mouse is not a human, right? And neither is a rat, neither are cells, neither are zebrafish. So that's really something important to keep in mind because we interact with the world, people as us, every day. And my initial research interests were focused solely on social behavior and understanding how the brain actually governs these interactions. And I wanted to understand that from even before birth all the way till advanced ages. And that's just that timeline right here. But genes in humans do equal, in some part, most part, I would say, genes in rodents. The question that we wanted to ask, though, was can we address some of these complex questions in the brain, or of the brain, if we manipulate those genes? Because it's not always the case, right? Again, I said a mouse is not a human, but these surrogate endpoints are crucial, I would say essential to getting therapies or therapies that we could test. And how do you relate this to Champ1 disorder, right? So now I'm going into some of the granular biology, which I'm trying to avoid, but I wanted to show you these slides because I think you may have seen them before, maybe, if Jeff shared the slide deck with you, that I prepared for him a couple months ago. And so it was a slide deck that showed where Champ1 was expressed in the brain, right? Because I wanted to understand, well, there really isn't much out there. I think there are probably less than 40 publications on Champ1. Is that correct? Maybe even fewer, right? Maybe 20. So there's this database and a number of databases that you could find online. Everything is online now. There's big repositories of data, especially for data related to what we call the expression of genes or proteins, right? There's one called the Allen Brain Atlas, and the Allen Brain Atlas is what's shown here, these 2 screenshots basically showing the brain of a mouse that was cut in half, and you're looking at it from the side, right? And what it's showing is expression of Champ1 in these 2 areas of the brain that are important for learning and memory and for walking and for motor. And this is just another way to look at it. This is a 3D map. And I could send you these as well, or Jeff can, basically showing where Champ1 is expressed. And these are instructions to understand how to read this kind of map. And that's what we've been trying to do, is just basically understand where we are. Because when Jeff came to me 11 months ago, he was a random guy calling me on my cell phone, or he called my, no, he emailed me first. I gave him my number. He called me on my cell phone. I wasn't even in Houston at the time. And he said, can we talk about this disorder my son has? I said, sure. I'm walking across the street right now, but I'll call you back as soon as I get back to my office, or the office I was staying in. I call him, and then he basically unloaded onto me, right? But that's exactly what you need to do. You need to be your advocate for you, within your community. And what I would say, for better or worse, be shameless, right? And basically just go out there, because you don't know what's going to stick. There will be people that you encounter that would be willing to be part of your team. Like I said, teamwork makes the dream work. You find that, I would say, strategic team that helps advance your agenda as a community. You're one of the 7,000 that are trying to do the same. So you find those individuals who are willing to work with you. Jeff was one of those people, contacted me, and I said, sure, why not? Let's talk. We talked about it, and it seems like it's working, right? So mice and rats. So how do you model mutations in them when they all look the same, right? So this is just a funny picture, just showing a mouse's face. That mouse might have motor problems or social problems, et cetera. They all look alike. So what we do is we examine the parts that make the whole, right? I used that analogy earlier of Legos, right? So this is what we call like phenotypes. If each individual Lego was a phenotype, the entire organism, you'd have to interpret that entire castle in that context, right? So I had said earlier, social behavior. I'm just going to show some videos that perhaps don't entirely, I don't know why this isn't working. Okay. May not make sense to you, right, when you're looking at it. But for us, as I would say, behavioralists, when we test animals in these different assays, so we call them assays, right? So just evaluations. We're trying to understand in a very simple way, when you have what we say a tractable organism like a mouse and manipulate its gene like Champ1 and put it in a test. So this number 1 is the partition test, or number 2, let's say this down below is a test for aggression. We call it the resident intruder task. We're trying to look for ways to interpret this behavior, right? By the time that the animals spend either investigating a novel partner or engaging in aggressive behavior, or in this case here, investigating whether or not the mouse spends more time, you're a test subject, with a novel mouse under a cup, or maybe another cup that has is either empty or has a novel object. So a lot of these, if you're looking at it for, I would say, more than what it's worth, then you're interpreting it incorrectly, right? What we're trying to say here is that this is one piece of evidence that would suggest or demonstrate, right, some evidence of social problems in this mouse model, but the way we interpret it. So we build upon the evidence, all of these pieces of evidence, by doing different behavioral tests and also collecting samples from the animals, so such as their brains or blood, plasma, et cetera, liver, kidney, different organs. And we look through sequencing methodologies, different ways to profile changes in their metabolites, for example, to find that entire picture, because nothing could be, I would say, interpreted well in isolation. You're probably wondering right now, where is that Champ1 mouse? So we currently have 3 groups. I don't have it listed here, but 3 groups are working on the Champ1 or creating Champ1 mouse models. So 1 on the East Coast, and then 1 on the West Coast, and 1 in Texas. So that's my group. And so those mice are in the pipeline to being available. There's one, I would say, that is more advanced compared to the others, and that's one that will be available broadly for wide distribution. and that's through the Jackson Laboratories in Maine. So they have told me that they have some positive, I would say very encouraging results. So hopefully, once those are released, they will, I would say, work with us as a foundation, and Jeff has this information as well, and work with the researchers who are interested in this mouse model to then distribute the mouse and then have them study with their various expertise, right? So I'm a behavioralist or behavioral geneticist and neuroscientist. I'm not a neurophysiologist, right? But I collaborate with those individuals, and I have them take the mouse, put metal in their brain, and basically do EEGs of mice, right? And that is another, I would say, aspect, is that collaboration, because biochemists may not know exactly how to work with, I would say, behavior, right? So they wouldn't walk into a behavioral suite and start testing mice immediately. Their bread and butter would be taking a tissue and then isolating protein, right? So it's basically relying on the expertise of the team to then discover what might be important biological pathways that could be potentially targeted for therapies, okay? So mice are not the end-all be-all either. And I'm showing this slide because mice and rats, cell models, those are all needed. This cross-species approach and using multiple model systems, that provides us as researchers with the tools for treatment. So there's advantages and disadvantages for all of them. I would say for mice, they're small. I would say they're very tractable in terms of expanding them. They're cheap, but then they are very skittish, right? So if you're trying to study anxiety, that's a very different model compared to using rats, which are, I would say, very behaviorally rich, and their repertoire of the behavior assays are completely different from mouse. There's some similarities, but they complement each other. Studying iPSCs or cellular models, right? So getting those fibroblasts, getting those, I would say, punch biopsies from patients, that's also something that's important, but then you grow neurons in a dish, you understand what might be expressed, other things that are modifiers or factors that are changing Champ1's function. But then it's a cellular model. It's a reductionist approach. It's not something that's made up of all the different neurons in your cell types of the brain, right? So these things all add up together and are needed to bring therapy to light. So this is basically showing some of the phenotypes that you guys have described today. One thing that we are trying to do, or we're proposing to do, is taking, using genetics to basically selectively remove Champ1 from the brain, to ask that specific question, is the brain really that important for all these problems that we see here? GI, autonomic dysfunction, sleep problems, cognitive problems, et cetera. So that's one way that we're going to try to learn about the biology. It's not a way in which we're going to try to understand necessarily a new therapeutic pathway, right? But it's to better understand if these problems that occur are actually coming from brain, right? These tools altogether help us understand the mutations as well in Champ1. So there are things as Aviana has a point mutation, a specific variant. So there are ways in which we can create these mouse models, and we're trying to do that now with Champ1 to understand if you have Champ1 completely removed, is the phenotype or the way that animal looks or it performs very different from a specific mutation in Champ1. And then by studying these mice together, that's how we're going to leverage these actionable therapeutic targets, even independent of the why, right? So when I say the why means like this biological mechanism. So why is it that the brain is causing these problems in GI dysfunction, right? Maybe that doesn't even matter because you'll have the model in hand and you could already start testing potential therapies. Does that make sense? So you have two camps. Both of them are working in parallel. The basic biologist trying to understand mechanism, as well as the preclinical, I would say, investigators trying to understand therapy. And those two are both needed to push the therapy forward. Oh, and here are the takeaway messages, right? So who? You all. And what these models, research, and the endpoints, and these biomarkers, outcomes, et cetera, are really important. There was a question of timing, and maybe that's something that we could discuss at some point later today, because the timing of intervention is equally important. There are other foundations which I work with, or other community groups, other genes, that the aspect of timing has become, I would say, a priority, because they have something that might potentially work, but what they're finding, and they need more evidence for this, is that perhaps the genetics is telling them they need to start intervening earlier on. But I would say the data are not complete, right? So this is why participating in research studies, being part of the conversation and engaging with the researchers and other members of the community, as well as with industry partners, is really important for, I would say, you all right now. And now, so you all right now, you're here, but for how long, right? So what I mean by but for how long is something I wanted to discuss as well. So once you put forth all of your eggs into this basket and commit to investing your time and effort, because time is something that you cannot put a monetary value on, right? And your children are growing up, they're getting older, and it is also, I would say, the bandwidth, your own physical time to actually do everything, work as well, but also maintain your relations, personal, interpersonal relationships, and also try to take care of your children. So that is where I say, but for how long? If you decide to make the commitment and you're here today, I really think that you should be in for the long run, because it's going to be a war of attrition. It will be not tomorrow or a year later or 3 years later. I'm hoping less than that, right? We all hope for that, but we have to be realistic and manage our expectations. And I would say for the researchers to not overpromise, I would say, a cure, right? Because that that four-letter bad C word of a cure has been something of, I would say, some of the issues that have come up in the past for why communities don't interact with researchers as much is because they've been burnt, right? Because they've heard the word cure far too many times, right? But now with gene therapy and some of these other approaches, it's getting much closer. So this is a time to be really involved. And where? Worldwide. I think the expansion of it, so the representation of people within Europe here or from other countries as well. I think it's only European, right? Is there anyone else beyond that? There's a Japanese family. Oh, okay. Okay, yeah. So I think that needs to be expanded as well, those efforts. So I am also part of the Association of University Centers on Disabilities in the US, and we are trying to expand all of our efforts internationally with other international communities to basically focus on the needs of communities with intellectual disability. And the reasons are limitless to do this. And how are you going to do that? By coalition building. The infrastructure is needed for researchers and for the community themselves to build, right? This networking, the resources, the capacity, that's all needed. And I think at the end of the day, the resources, when I say resources, I'm talking about funding, is particularly important for the researchers. Now, Jeff could probably talk to you about this, talk your ears off about this. He probably already has. But he could tell you that that is probably one of the #1 factors to push research as quickly as possible. And that's my last slide. Anyone have questions? Yes?
I have a question.
Yes.
Once the mice are created, how do we get a phenotype? How expensive is that to get a phenotype? And is that funding that we apply for grants or we get through your foundation?
Did you say expansive or expensive?
How expensive and expensive?
Yeah, so I've agreed to already phenotype the mice. So I've talked with, so the mouse model that's being made by the Jackson Laboratories, that is made through a discussion with the Orphan Disease Center of University of Pennsylvania, right? So in that discussion, I said, if you give me the mice, then we'll phenotype them. Now, we won't phenotype them till the cows come home. What we're going to be doing is phenotyping them with assays that we think are the most, I would say, valid or relevant to the disorder, right? Or some of the conditions, I would say, of the children. it's not going to be everything under the sun. So what we want to do is provide a carrot, right, for the community, the research community, to say, here is a mouse model that's been made. Here's an investigator who's doing behavior all the time, day in, day out, who's already done behavior on a ton of other models with other foundations. Take it, right? So it's for other people to see, oh, there's this mouse model. Okay, this is an interesting gene because not only is it potentially a transcription factor, right, that it could bind to DNA and cause, who knows what, but it also is involved in mitotic spindle assembly, right? So it's like getting two different camps of researchers that might be interested in Champ1, right? So that's what we're going to do.
And how do we find out who those researchers are?
Good question.
Do you know who they are?
Wait, you mean where they're going to come from? So this is where you're having to get out as the family. So the community needs to work together to enhance the PR, right? And as much as I am not the biggest fan of social media, that's what people use these days. They use social media, they have publications, they have things such as strategic roadmap white papers, right? That's what they're called, in which communities say and outline in a very short document, maybe 2 pages at most, this is what we want. We need mouse model, we need rat model, we need something, 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 right? Just outlining, bullet pointing that. And then working with NIH, so federal, I would say, foundations, so other foundations that are more umbrella foundations, as well as federal funding agencies, and advocate on your behalf, right? So say if you know a researcher who knows somebody within NIH, right? And you said, okay, please work with me to engage that individual at NIH who is a program officer and pitch Champ1 as something to focus on. That's what you need to do. You need to be, I would say, not just assertive, but you have to be aggressive to get things on the map, especially among these 7,000 rare genetic disorders with a gene, a protein, Champ1, where there are fewer than 30 publications. It's tough.
Yeah.
I'm speaking like it's a cakewalk. It's really not, because I've seen this before with other foundations, other genes, and there's a lot of enthusiasm to get it to start, and then just fizzles out, right? And I'm hoping that that won't happen here.
I have a question. I'd like to pick off where you left off. I've dealt with research before, and your very first slide, you had phase 1, phase 2, phase 3 with sample size. My question to you is, before you release any kind of information to us, how big of a sample size do you need? How confident are you? And if you're not that confident, will you be using any kind of smoothing data technique to give sort of like favorable report with high marginal error? Those are things I run into quite oftenly in order to get funding out of people. So I was wondering if maybe you could speak to that line a little bit.
So rare genetic communities actually fall under a different category from what I understand. I don't deal with the human data itself, right? So I'm a basic scientist, but if Wendy was here still, maybe she could address that question. But you're saying what is the minimum criteria needed to move something forward? Right.
Or whatnot. A lot of times what happens is we have a very small sample size and, well, we don't have a lot of confidence in that, in the data we're seeing. Your phases on your slide demonstrates that the higher the sample size, the truer, the more confident we are about the statement we're making.
Right.
A lot of researchers tend to smooth their data to make it look a certain way or whatnot, and a lot of times it's just to kind of push a certain direction or not.
Right.
I just wanted to know, making sure that doesn't happen to this group.
Right. So the academic researchers working with industry partners, you'd have to find the right people, right? So what happens when you're doing phase 1, phase 2, phase 3, since you've worked in that space before, you're working with pharmaceutical companies at that point, right? And they're the ones who are running the clinical trials, not the academics, right? So then, let's say Alzheimer's. I like to use Alzheimer's because you hear it in the news. It's almost like every 6 months, some big blockbuster drug that's in phase 3, suddenly, Biogen loses money or something because their drug didn't work out, right? You just heard about, that was one that's very recent. So I'm using that as an example. And I think in that scenario, I don't know personally, actually, of the information that led to phase 1, phase 2, phase 3, right? Because I don't really know what had happened with that clinical trial. Rare genetic disorders, as I said earlier, that is becoming apparent, I would say totally obvious, that you're going to find individuals or communities which there are only 40 or less, right, individuals. And the FDA has now taken that into account. And they're saying that patient advocacy and the voice of the patient is going to be really important to bypass some of these criteria that are required for more idiopathic or general diseases, right? Diabetes, let's say, for example. Does that make sense? Okay, yeah. Natural history studies are also really important. So Simon's VIP, or what is it? Sharelight? What is it? Searchlight, right? Searchlight, Spotlight, yeah. So Simon's VIP, since that's what I'm used to always saying, Simon's VIP is really important to get your data in there, right? That's another way in which you could, I would say, garner attention from the community. Because a lot of researchers, whether they be basic scientists or physician scientists, they know about Simon's VIP and they know about all of the data that are in there. So it's important to engage these types of umbrella organizations that are working with a number of different, dozens of different rare genetic disorder communities. Yeah. Oh, no questions, okay.
I think we have time for maybe one more question, if anybody has one. All right, Dr. Samaco, we thank you so much for everything.